Okay, so sorry for the hitch. I hope everybody can hear us. Yeah, really the issue here is to try and just understand really what's going on in terms of understanding vaccines, but also in terms of really understanding in particular the COVID vaccines and the issues that are beginning to emerge. So now, Marianne, are you, what we want to do today is really to look at vaccine research and development landscape, but in reference to SARS CoV-2 vaccines. Also look at vaccine dosing and schedules, again in reference to SARS CoV-2 vaccines. And then really look at the current debate and the current science surrounding matching and mixing of SARS-CoV vaccines. We will go through this. I will do the first couple of slides, and then Daniel will come in, and then eventually Marianne. Next slide, please. Now, just in terms of Marianne, this is a wrong slide, I think. Just go back a bit. Okay, then the one. Yes. Sorry. In terms of just understanding where we come from, been looking at prime and boosting vaccines. Too many that we are talking about this now, but this has been going on. Really, the whole point here is that when you get a naive individual at day 0 and you give them a primary vaccine, a prime, then usually it takes almost 7. It's usually slow and not very strong. And then over time, that immune response begins to decline. At that time, we can look for antibodies, we can look for memory cells, but even this prime, to some extent, has ability to protect either from disease or from infection, just depending on how the vaccine was designed. And then you come several days or several weeks later with a boost. Usually that boost will give you a very quick response. It will be rapid, it will be much stronger, and it will also give you a prolonged protection. And really what you are looking for here is to boost the memory cells that were initially primed by the primary vaccine. So prime and boost really to look at these aspects of ensuring that you have a second response with a much wider level of immune response, and also a much wider and a more quality T cells, B cells, as well as memory cells. Next slide, please. And then in terms of just understanding the vaccine platforms, your traditional vaccines have been things like live attenuated vaccines, inactivated vaccines. But now the emerging platforms include the RNA and DNA vaccines we refer to as nucleic acid vaccines, also includes the viral vectors, and also includes the recombinant proteins. So nonetheless, those are the emerging platforms of the vaccines we are beginning to look at. And as we proceed with the talk, we shall go a little bit more into details at how these vaccines work. Now, I've lost the slides. Marianne? Marianne, you are sharing the slides, please.
Let me upload them again. I thought that you can see
Okay.
them. 1 minute, sorry.
Okay, great. So let's go to the, all right. And then the issues of, we have finished the platform. So the issues of, go to the next one, next? Yes, the next one. Now, vaccine research and development is a very important process. To let you know, we've been researching on HIV vaccines for a very long time. And we normally start with a concept at laboratory. We design the antigen. Then we test the antigen in mice, preclinical. We also test that antigen in non-human primates. And then we move to phase 1. And then we continuously then move to phase 1 or phase 2, or we can move from phase 1 to phase 2B, and eventually to phase 3. So you will actually find that majority of vaccines hardly ever go beyond phase 1 because they end up being safe, but they are never immunogenic enough. And these days, really, we are moving from phase 1 and we move an expedited review to phase 2B. Phase 1 is when we are really interested in immunogenicity because you are giving, safety because you are giving this vaccine for the very first time to humans, and a bit of immune response. And then when you move either to phase 2 or phase 2B, then phase 2B is really an expedited process where you look at 3 things or 4 things. You look at extended safety, you look at immune response, you look at dosing, you look at the schedule, and to some extent, you look at also efficacy. And these are the current aspects they call proof of concept. And if this proof of concept works, then you quickly move into phase 3, So this avoids the long process of moving from phase 1 to phase 2 to phase 3. Next slide, please. And then looking at it in a little bit more detail and trying to understand the story where people keep saying that the COVID vaccine was developed very, very fast. The traditional approach is usually exploration, where we are doing research and discovering antigens, where we are looking at research and discovering correlates of protection in humans, and at the same time developing the antigen to begin preclinical work. This is just a investigation of new products. And then if this aspect of exploration works, then you move first in human. And usually the traditional approach has been to move from safety, phase 1, to look at safety and immunogenicity, phase 2, and to look at safety, immunogenicity, and efficacy in phase 3. And then if all goes well, then now you begin to go into applying for licensure, you begin manufacture, you begin marketing, and then you do phase 4, which is actually effectiveness in the real world. But really what has happened with the COVID is that the process has been expedited. So we have actually used what we call pandemic accelerated model, where research was ongoing, and as new antigens are developed and correlates of protection are realized, preclinical work begins. And as quickly as some preclinical data come in terms of safety, then this is quickly pushed into phase 1 and phase 2 and phase 3. And as phase 1 and phase 2, and to some extent phase 3 is going on, they didn't even wait. They began manufacturing. And then once there was good data from phase 1 and phase 2 that there was good safety and good efficacy, we were given globally what we call authorization before even the licensure was actually put through. So as we speak now, actually there is emergency use authorization ongoing, but at the same time, there are various clinical trials for some of these drugs, the vaccines that are under emergency use authorization. Phase 3 are actually ongoing. At the same time, phase 4. That is really the reason why the whole aspect has been expedited. And then when you look at what is going on, ladies and gentlemen, this is unprecedented. As we speak, there are almost 180 vaccines under laboratory and animal investigation. And then we have almost 30 vaccines undergoing safety, phase 1. We have almost 34 that may be getting into phase 2. And then 28 of them are getting into large international trials. This is phase 3. We have about 17 vaccines that are currently being offered in general population. All right? And about 8 are being given maybe under emergency use authorization. So this is totally unprecedented, and that's why there is very competing nature of ensuring that the clinical trials are going on, data is being collected, and at the same time, emergency use authorization is on. And now to even complicate matters, we are now beginning to talk of mix and match. The next one. And then the whole issue of the dream vaccine. What will be our dream vaccine? The crown jewel antigen is actually the spike protein. There are two aspects with the spike, the S1 and the S2. And these are fused. So the first one binds, it is cleaved, and that enhances it to bind even further and have access into the cell. As you are aware, viruses are intracellular, are obligate intracellular microorganisms. But what confronts us on this crown jewel is availability from variable to conserved regions. So really where the science is now is trying to understand, are there really conserved regions that are very critical conserved region to make the vaccine? This actually will be a universal vaccine. The other dream aspect of it is what we call a chimeric spike. A chimeric spike would actually be an mRNA platform that then contains spike genes sequences from various SARS CoV spike proteins. Can be SARS CoV-1, SARS CoV-2 viruses to produce a mixture of proteins, domains that may then confer a very broad analyzation for both T cells as well as antibodies. This is traditional whole virus, right? Completely inactivated. But if you're going to whole virus inactivated, then definitely you will need a cocktail to ensure that you take care of the variability. And then on this other side is to look at science and put together all the receptor binding domains of SARS-CoV viruses. And then within these, put them in such a way find and form a known, but it will contain all the aspects from the Delta, from the Gamma, and as well as from other SARS. You want to prevent now as well as in the future. And then the other aspect of a dream vaccine with serial vaccines. One pan-coronavirus approach where you actually deliver a series of different spike proteins. All right, this you're actually immunizing for nearly all the variables that come in terms of variables that you find in the spike protein. This is actually the discussion that is going on, and this is where science is trying to take us, so that eventually one of these will actually form the dream vaccine. But the ask of that dream vaccine is to ensure that we take care of all these particular variants. Next one. And this is where the problems are, the variants of concern, where we have issues to do with heightened transmissibility, more severe disease, reduction in neutralization, reduced effectiveness to treatment of vaccines. And then we have variants of interest, where really we know the variant exists, but we don't have the epidemiology to go with it in terms of trying to understand whether it is more transmissible, whether it is actually a neutralization from natural infection, as well as vaccines, whether it is actually causing severe disease. So these are variants of interest. I'd like to draw to you the fact that all the variants of concern, the alpha from the UK, the beta from South Africa, the gamma from Brazil, the delta from India, realize that all these variants emerged in 2020, September 2020, May 2020 for the beta, November 2020 for the gamma, October 2020 for the India. And then also realize that these variants of interest, Eta, the Lota, the Kappa, and the Lambda, again, all emerged last year, December 2020, Lota, November 2020, Kappa, October 2020, Lambda, December 2020. So what is this telling us? We are now into July. The pandemic has not gone away. And for this year, we have not isolated any new variant of concern or any new variant of interest. So what does that mean? And that is a question I want to leave with us to discuss, that all this have emerged immediately last year when there was heightened transmission. The virus was spreading globally all over the place. But come 2021, we are not actually seeing any new variants of concern or any new variants of interest. So I will, and I'll call on the Dr. Mwema to take us through the next slide.
Thank you, Prof. Anzala, for that piece of information. Yeah, so I'm going to take us through what it takes to take a vaccine from development all the way to licensure, mainly looking at the initial developmental stages of vaccines and also the determination of dosing and schedules with reference to SARS CoV-2 vaccines. Next slide, please. Yeah, so there's a lot of work that goes into vaccine discovery or vaccine development long before even preclinical and clinical studies are initiated. And this is mainly exploratory work. For instance, when you have a new pathogen which has emerged, you'll want to know what kind of proteins it has, which can be used as targets for as vaccine as vaccine immunogens. So things like sequencing the genome of the virus or whichever other pathogen has emerged, those are critical for development of a vaccine. And also many other things like pathogenesis, all those inform the vaccine design. And for instance, here I'm showing a slide which is showing how SARS CoV-2 spike protein was identified as a potential vaccine antigen. In this particular study, they were able to take people who had just recovered from COVID-19, and then they were able to get their blood samples, and they were able, through a series of steps, to isolate monoclonal antibodies. And by doing some PCRs and cloning the immunoglobulin genes from the B cells that were activated in this particular individual. And then those monoclonal antibodies were then characterized and identified to be neutralizing to the virus in vitro. And then they were able to also map the epitopes that were targeted by those neutralizing antibodies. In this case, the ones that were neutralizing were identified to be targeting the spike protein. And then to test that further, then they took mice that they gave these monoclonal antibodies, and then they challenged the mice with the SARS-CoV-2. And then they were able to monitor the viral loads in these mice over the days and sacrifice the mice at day 5. And then they were also able to monitor things like weight loss and just pathogenesis of the mice. And so you can see at the bottom right corner, the control mice had detectable viral RNA in the lung at day 5, while the mice that got higher doses for the monoclonal antibody, this particular monoclonal antibody, the ones that got higher doses were, they had significantly lower levels of viral loads, SARS-CoV-2 viral loads in their lungs. And that was dose dependent. So this is one of the many studies that were done to identify the spike protein as a potential vaccine antigen. Next slide, please. And then once those exploratory studies are done, then what people did, they went into preclinical studies in mice. Mice are usually easy to work with because they are small and you can get large numbers, and they're much easier to handle compared to larger animals and compared to humans, of course. And so this is showing some of the preclinical work that was done in mice for the Oxford AstraZeneca vaccine, the ChAdOx1 and COVID-19. And in this case, they took mice, two different types of mice, the BARB-C mice and the CD1 mice. And then for some of them, they gave the COVID vaccine, the one that is shown in circles, and the others, they gave the control. So basically, they didn't get the vaccine antigen. And so you can see that the mice that got the vaccine antigen actually generated neutralizing antibodies. And also when you look to the far right, there was a tendency of the ones of the two groups that got the vaccine antigen to generate T-cell responses. And that was the same for both mice. So this was a clear indication that this particular vaccine construct was immunogenic, at least in mice. Next slide, please. And then what would be done after showing immunogenicity in mice is to go to more complicated systems, bigger animals, and usually the animals of choice are non-human primates because they are closer to humans. And so in this case, the same group decided to test the vaccine construct in rhesus macaques. And then if you look at the left side of the slide, they were able to show that this vaccine, when it is given either as a prime or as a prime boost regimen, compared to the monkeys that got the GFP, the ones that got the vaccine either as a prime or a prime boost regimen were able to generate neutralizing antibodies. And also they are also the same ones that were able to generate T-cell responses compared to the greens, who are the negative controls that didn't get the vaccine construct. And so they also, the good thing with non-human primates is it's also easy to test the ability of the vaccine to protect from challenge with the virus. And so they took those monkeys and challenged them with the SARS CoV-2. And then they monitored their clinical profiles to try and delineate the monkeys that developed pneumonia-like symptoms. And so from the middle graph, you can see that the green monkeys that didn't get the vaccine construct generally ended up with a worse clinical score compared to the red and the blue monkeys that got the vaccine. And also they further characterized the quantities of viral loads in these monkeys in the bronchoalveolar lavage. And you can see that the green monkeys are the ones that are detectable viremia compared to the blue and the red ones. So this was at least a proof of concept that this vaccine is immunogenic and prevents SARS-CoV-2 pneumonia in rhesus macaques. Next slide, please. And so after doing those preclinical studies, that's when the vaccine enters the clinical stages. And as Prof mentioned earlier, this usually starts with the phase 1, which is mainly meant to look at safety. And also in some cases, dosage can also be evaluated by doing dose escalation studies. So in this case here, we are showing the evaluation of the Pfizer vaccine, the BNT162B1 and B2. Here at this stage, they still add 2 variants, one which is soluble and another one which is membrane bound, variant of those 2 mRNA-based vaccines. And so they actually wanted to use this phase 1 clinical trial to identify which of the 2 is better with regard to both safety and also which one had a favorable immune response based on different doses. So when you look at the left side of the slide, they evaluated the safety profiles for the 2 vaccines, both the B1 and the B2, the B1 at the top and the B1 at the bottom, after the first dose and also after the second dose. And they were able to show that there were no life-threatening side effects in this case. All the side effects were either mild or moderate, and just a few severe but not life-threatening, not requiring any hospitalization. And then on the right-hand side, where they were actually looking at the immunogenicity for the different dosages, where they compared 10 microgram, 20 microgram, and 30 micrograms, they were able to see that generally 20 micrograms and 30 micrograms were good for either of the 2 vaccines. And then, but generally 30 micrograms were actually giving an early initial response at around 21 days at the point at which they were giving the boost compared to the others. And so in this case, they decided to choose the 30 micrograms going forward. Next slide, please. So this is another study that was done for the Oxford AstraZeneca vaccine. Here they actually combined a phase 1 and a phase 2, whereby they started the trial by first immunizing a small group of people without blinding. It was an open, the phase 1 part of it was open labeled, and it had only 10 participants who were followed very closely. And then they extended the vaccine to make it to include phase 2 and phase 2 aspects to it. And this is showing the safety profiles that were seen in the phase 1 arm when they were closely monitoring safety for this Oxford AstraZeneca vaccine. And when you look at the key, you can see that, and you look at the side effects, you can see that none of the individuals that were evaluated required hospitalization. Actually, most of the side effects were moderate or mild. And they also evaluated many others that we didn't show here because of space. But generally, the vaccine was had a very good safety profile, and that was quite encouraging. And then another aspect that they analyzed in this particular clinical trial was to look at the dosages and also the scheduling. So this is how vaccine scheduling, especially the intervals between doses and also the number of doses are determined. And so here they add the arm that was boosted at 28 days. They got the prime at day 0 and then boosted at 28 days. And then they were able to show that boosting was important because they could see an increase in response beyond 28 days after giving that booster at 28 days. And then the other question that came up was whether you really needed 28 days or whether due to the logistical constraints that the world was having because of poor availability of vaccines, would it be possible to space the dosages much more so that you can give the boosting much, much later? And so they happened to have two groups actually, that received the booster dose at 56 days. And actually one of those two groups was receiving the standard dose, which had been given in the other group, which was boosted at 28 days. And then the other group was actually receiving a lower dose. And they were able to show that, especially for the standard dose group, boosting later does not compromise the quality of immune response. When you look at the immune responses that were elicited at the end of the day, they were comparable to the immune responses that were elicited after boosting at 28 days, suggesting that if you give people the standard dose and you, but you space the doses a little bit more than 28 days, you still get a very good immune response. Next slide, please. And then, so after those phase 1 studies, the next thing was to try and expand this analysis, because phase 1 studies usually tend to restrict the populations of interest to people who are fairly young, healthy. Yet phase 2 studies, you can easily evaluate more age groups. So in this case, they decided to expand the analysis to include people who are much older, all the way to people even who are over 70 years old in this phase 2. And they also add a larger number of participants to be able to determine also safety and also immunogenicity and also dosages. So in this particular case, they were able to show that the vaccine was safe across all age groups. And also when they looked at the immunogenicity also across all age groups, for people who got the 2 doses across all age groups, when you look at the graph on the right, they all had comparable immune responses regardless of age. And only the people who got only 1 dose had lower responses, still emphasizing the fact that 2 doses were required, and that regardless of the age of the participants. Next slide, please. So, but then something else came up, and I mean, the dosing intervals that are usually evaluated in vaccines have been determined mainly historically and based on the best estimates that the scientists have at the time when they are designing these clinical trials. But then, because of the complex nature in the complex environment in which these vaccines were being tried, and also because several phase 3 clinical trials were also started, and there were many participants that could be analyzed, there were some people who ended up getting their booster doses much later, just because of logistical reasons. And so they took advantage of this to try and see whether even spacing the doses even further could improve immune responses, or whether it could compromise immune responses. At that time, of course, they didn't know, and whether it could also even improve efficacy. So they took advantage of those logistical problems and decided to evaluate the impact of delaying the second dose in various groups of participants. So on the far left, the analysis showed that when you actually space the first dose and the second dose by more than 9 weeks, you actually even get a better immune response, regardless of whether those people are in the low dose group or whether they are on the standard on the standard dose group. And the same actually was even observed even with a much better immune response after spacing the 2 doses by 12 days. That was observed for people who are between 18 and 55 years, but not in the much older people whose immune responses remained the same regardless of the spacing of the doses. And in fact, even when they analyzed the efficacy, as in the ability to be protected after the second dose, they actually saw that those people whose doses were delayed up to around 12 weeks, even that those groups tended to show better efficacy. So suggesting that much more would have been missed if this secondary analysis was not done, and if we had only stuck to the usual dosing spacings that are usually anticipated in clinical trials. Next slide, please. Yeah, so by the time these vaccines, as Prof mentioned, were being given emergency authorization, most of them are still having phase 3 clinical trials ongoing. And in fact, there's some data that we haven't shown because of time that was coming from phase 3 clinical trials that were still ongoing, and also the phase 2 clinical trials. And those are the data that were actually used to determine the efficacy of these vaccines. And something that has come out over the year is that both T cells and B cell responses contribute to vaccine efficacy. And more evidence suggests that to protect from absolute infection, to give that absolute protection from infection, neutralizing antibodies play the more prominent role, as seen in that graph, which is on some work that was done in Australia, where they did some meta-analysis across all the vaccines. And they were able to show that the efficacy, the protective efficacy that has been reported for various clinical trials, based on all the vaccines that are in development or that have been recently developed, strongly correlates with the mean neutralization antibody levels. So emphasizing the importance of neutralizing antibodies in protecting from infection. But then we also have additional evidence that is suggesting that T cells, they might not necessarily prevent infection, but they could be important in preventing disease. And actually, they might even be able to prevent disease even when new variants emerge, unlike antibodies that tend to be quite strain specific. Next slide, please. So this brings us back to this slide, where Prof mentioned all these variants of concern and various variants of interest. And actually, some of these variants have been defined as variants of concern because they are resistant to neutralization. Neutralization that has been elicited either from natural infection or neutralizing antibodies that have been elicited from vaccine, from vaccine in people who have received vaccines. Next slide, please. So with those variants in mind, it has actually been shown, as I mentioned earlier, but just to show you a little bit of those data, that the new variants are likely to escape antibodies, but they are much, much less likely to escape T-cell responses. Some are emphasizing the importance of eliciting good T-cell responses because those might give pan-variant protection. So in this particular study, what they did, they took people who had never seen COVID-19, who had never been infected with SARS CoV-2, but received a vaccine. And then they looked at the antibody responses at the first time, just after receiving the first dose. I think this was 14 days after receiving the first dose. And also the next column shows 14 days after receiving the second dose. And they were able to show, which is more clear in the 14 days after receiving the second dose, the second yellow column, they're able to show that while these people were able to neutralize wild-type variants, the wild-type variant, the emerging variants, some of them, especially the South African variant, which is the third column within that big column, they were more resistant to the neutralizing antibodies that were elicited in these people. And then the blue people, the blue dots are showing another cohort that consisted of people who had been infected with COVID-19. And then they ended up receiving also the first dose and the second dose. So in the column which is showing these people after recovering from COVID-19, this group were able to show that the neutralizing activity of their serum was much lower when it was tested against the South African variant, which is in the third column within that section, compared to the wild-type vaccine and compared to the UK variant. And also the same was also observed for the, after these people were given the first dose and the second dose, whereby the new variants were more resistant to neutralization. But when you look at the graph to the right, where they looked at the T-cell responses, And they tried to activate the T cells of these individuals with the peptides from these variants, with both and also from the wild type virus. Even for the people who had just received the vaccine and had not been infected with SARS CoV-2 previously, there was no difference between reactivity to the variant and to the wild type, to the original virus. And also, even after receiving the vaccines, there were no differences between the two reactivities, either to the variant or to the wild type viruses, suggesting that the T cell responses that are induced, whether it is by natural infection or whether it's by immunization, those T cell responses tend to cover. They are more likely to cover across variants and give pan-variant immunity. Next slide, please. So with that, that's how we have ended up with all these COVID-19 vaccines, as Prof mentioned earlier, where we already have 17 vaccines that are currently being offered to the general population, and also actually 8 vaccines that are in wide use across the globe. And with that, actually, that gives us a chance to even go to the next phase of this presentation, where Marianne will be talking to us about the mixing and matching.
Thank you very much, Daniel. So yes, as you have just mentioned, this next section we're going to just talk about mixing and matching of vaccines. Really, there's now this new debate: can we mix vaccines? I.e., if we give the prime, for example, AstraZeneca, can we come back later and boost with Pfizer? Is it safe, and will it elicit maybe even a much stronger response? So this is ongoing conversation and has elicited quite a bit of debate. And this is what is being called heterogeneous vaccination, or in other words, mixing and matching of COVID vaccines. Again, as my colleagues have mentioned, we have all these vaccines that have been licensed for emergency use. And you can see that in various regions across the world, we have different vaccines. Like, for example, within African region, we have AstraZeneca vaccines, which sometimes are available and sometimes they're not. And the big debate that has led to this mix and matching conversation, if this AstraZeneca is not available, can we then give a second boost with, let's, for example, say an mRNA vaccine such as a Pfizer or Moderna? Again, as I mentioned, it's an ongoing conversation, quite interesting, in that a few days ago, we saw that WHO actually giving a statement that they're quite cautious about mixing and matching of vaccines. In fact, they just keep emphasizing that we should give the same vaccine, again, because we have paucity of data. We don't have enough data that can actually boost or say that indeed we can mix and match vaccines. And again, you can see all these news agencies, but even as you're talking about this, some countries such as Germany, France, and even Spain have already started mixing their vaccine. Here we saw a few weeks ago, Angela Merkel actually mixing a vaccine. She got her first shot, AstraZeneca, and then later on followed by the Moderna. So really here and in this discussion, we want to really interrogate the data. Some groups have already started investigating to see, indeed, do we get a good immune response if we mix and match, and is it safe? So again, this is just a preamble of what mixing and matching is, and again, it's referred as a heterogeneous prime boost vaccination. And again, what led to this, first of all, as I mentioned, having the availability of these vaccines. Sometimes we have AstraZeneca available, sometimes we don't. So is it okay to mix? That's one thing that led to this mix and match. And in the European countries, what brought up this debate is this rare events that started or were reported, particularly after using the AstraZeneca vaccine, leaving people partially vaccinated, a bit hesitant about getting a second boost of AstraZeneca. So again, these questions came up: would we mix and match these vaccines and see if we get an even much stronger response? Again, this debate is not new. It has been seen and proven in other models, in other vaccines. For example, in HIV, you remember the trial, the Thai trial that was done almost 8-9 years ago, phase 3 trial that showed that this HIV vaccine was 30% efficacious. What they did a mix and match where they first gave a prime using a canary pox vector, followed then by 2 boosters of a recombinant glycoprotein vaccine. Again, it was safe. And again, this is still ongoing conversation, research on an effective HIV vaccine that indeed will use this mix and match to elicit a more better immune response. This again has been seen in the Ebola vaccine that is currently in use in Western Africa. And I have to emphasize that this Ebola vaccine was tried here at Kavi, and we have this publication where they also showed that successfully using the first shot of an adenovirus that was used, and then followed with a booster shot of an MVA vector, elicited quite effective, quite good immune response and was safe. So again, this is not a new debate, has been used historically to study other vaccine models. And again, it's good to see that it can be used and can be quite efficacious. And with that, then what now the science is looking at, indeed, with these COVID vaccines that we have a mixture of vector vaccines, we have mRNA vaccine. Can we mix them? Are they safe? And as I keep mentioning, will they give a good immune response? So what we have 3-4 groups. Again, these groups are found in the European countries that have studied a cohort of people who have these mixed and matched vaccines. And in their model, they have a group of a cohort that received the AstraZeneca vaccine at day 0, and then primed 8 to 9 weeks later, using they primed with the AstraZeneca vaccine, so a matching vaccine that, boosted with the AstraZeneca vaccine. And then we had another cohort of people that then used the Pfizer or mRNA vaccine to give the boost. And then what they did is then study the different immune responses between these 2 groups to see if there was some good immune response following boosting using these different models, and 2, if it was safe. So in this study that was done by the University of Oxford, you can see that we follow the x-axis in this graph. We see that in a model, they boosted, first they gave the AstraZeneca vaccine as the first shot, followed then with the second shot of either the AstraZeneca vaccine or the mRNA, and in this case was the Pfizer. And they then tested against different variants in this model. And you can see that indeed, when they gave a booster using the mRNA vaccine, the cohort elicited a much stronger response compared to the more homologous AstraZeneca followed by AstraZeneca vaccine. And this was quite significant and across the different variants. So this is quite interesting to note and to see, and also gives a good confidence that mixing and matching the vaccine, and in this case, where they first gave the prime using the AstraZeneca, followed by the Pfizer, elicited a much more immunogenic response compared to the more homologous combination. In the second group, this was a study done in Sweden by the Karolinska group, where they then used again the AstraZeneca vaccine as a prime, and then followed up with the Moderna, that's another mRNA vaccine. And again, you can see across the board that indeed higher neutralizing antibodies, again showing a more immunogenic response when we mix and match the AstraZeneca, the vector vaccine, followed by the mRNA vaccine. You can see that the response elicited, and in this case, in this graph, you're looking at the neutralizing capabilities. You can see that boosting with an mRNA vaccine elicited a much stronger response compared to the matched vaccine given in this case AstraZeneca followed by AstraZeneca, and these responses were measured a few days after giving the boost. So again, really making a strong case that actually mixing and matching can give quite a strong immune protection or a much stronger immunogenic response, and in this case, the neutralizing antibodies compared to just giving a matched vaccine model. Again, this is continued from that group where again, they were looking at across different variants. Again, you can see that a much stronger immune response is elicited when you do a mix and match compared to the more matched or homologous vaccine combination. And again, here in this graph, we see that was a significantly better protection elicited against the different variants. In this case, they tested against the beta variant when they gave the more homologous or heterogeneous vaccine combination. Again, making a big case that actually mixing vaccines, and in this case, the vector vaccine followed by the mRNA vaccine is eliciting a much stronger response. Yes, we are waiting for more studies. And again, we're hoping we can do some of these studies as well in our setting, where not just starting with the vector vaccine, there are studies now where they want to see if starting or giving the prime of an mRNA followed by the vector vaccine, would we see a similar pattern? Again, giving us the confidence that in areas where they cannot get a certain type of vaccine, they can use the next available one, either to give us a prime and most importantly, as a booster. So we've answered the question, or so far the data that we have so far from the 3-4 groups that is studying mixing and matching of vaccine, we've seen that it's immunogenic. But the next question that we're asking ourselves, is it safe? So again, from the different studies, again, this was a study done by the Oxford group where they looked at the safety or the reactogenicity across the different cohorts that were tested with this mix and match method. You can see that again, this is just the boost. So just to make work easy in the presentation, everyone was given the prime boost and then given the prime and then boosted with the mRNA vaccine. And this is the Pfizer. And you can see across the different reactogenicity that they were testing, you can see that it was quite from mild to moderate with very few cases of severe and actually very few or very rare cases of hospitalization once this mix and match was done. And this was across different parameters that they looked at, both a local and systemic safety profiles. So again, really making the case, in fact, mixing and matching is quite safe. And you can see that these reactions or these safety profiles, the cohorts were studied across for a few days, for almost a week. And you can see any local reaction that developed following mix and match between day 5 and 7, any local response, any swelling had drastically reduced, and any systemic reactions such as headaches, fever, etc., and also by day 5 to day 7, cleared quite well. So again, making the case that first it's quite, you see a good or even better immune response, and also it's safe to mix and match this vaccine. So in conclusion, in summary from this data that we have seen so far, when we do a heterogeneous mix where we have the vector vaccine, in this example, the AstraZeneca, followed by an mRNA vaccine, we see a much stronger immune response, i.e. from the data we have seen so far, we see a much stronger humoral immunity where enough antibodies are elicited with good neutralizing titers and also spike-specific B cells. We also see a much stronger cellular immunity where we see spike-specific CD4 and CD8 T cells, and this is across the different variants of the SARS CoV-2 virus. So this is quite good news, and we hope to see many more studies that we look at these different combinations and study this further. So moving forward, as I mentioned, we need to really interrogate the type of immune response elicited and how long this response will last. Will it last quite, will it be a lifelong response? The studies now that are showing if we can also do a booster with a different vaccine. So really mix and match to see how this response will evolve over time. Then we need to keep a close eye on the safety profiles because we want to make sure that it's safe with less severe cases. And also, we need to be able to continue studying the broad immune responses and their potency towards these variants. So in conclusion, a mix and match approach towards these COVID vaccines may actually provide a more logistical relief where any vaccine that is available at that time point can be given either as a prime and most importantly as a booster. And we have actually seen some immunological benefits in this mix and match model. So over to you, Professor Nzala, to take us through the work that you have done so far in regards to the COVID vaccine. Thank you.
Thank you, Marianne. Thank you, Daniel. This is where we are now. What I would like to conclude with is just to show a bit of local data, a window into the local data we have actually started gathering. And we are conducting a study to understand immune response dynamics of Kenyans to the ChAdOx vaccine. Next slide, please. So just go back. So again, just go back. No, next slide. Next one, yes. It takes us back to, then the other one, yeah, it takes us back to this aspect that we really want to understand the immune response dynamics in terms of understanding in our local population, really what happens at the prime. When we pick samples at day 7, we pick samples at day 14, we pick samples at day 28, and then we again begin to pick samples when these individuals get the second dose. And our interest is really to look at both those who had COVID and got vaccines, and those who did not have COVID and got vaccines. And just to compare how is immune responses in these two different cohorts. And what I'd like to say here is that this study at CAVI is actually ongoing. And if any of you wants to join this particular cohort so that we can actually begin to understand our own immune responses, are totally welcome. Next. So this is just to show you so far, the data that we have collected. The individuals in red, these were ex-COVID individuals, where we picked samples at day 0 before the vaccine. And we were using the SARS CoV-2 IgG detection system. And this detection system looks actually at receptor binding, domain antibodies that bind to that aspect. So you can actually see that the ex-COVID individuals prior to the vaccine and because of exposure already had antibody levels way above detection. And when you look at naive individuals, it's interesting to also note that at day 0, there were quite a number who really didn't have much immune response to the receptor binding domain at day 0. But interesting enough, we had quite a number of individuals who already had very high immune responses. This could actually indicate individuals who got exposure, but really never came down with any particular disease. Because when we took history of these naive individuals, we were asking, have you actually suffered from COVID? And all of them said no. And yet, when you look at immune response, you can actually see in some of them, there was actually quite heightened. Then at day 7, you can actually begin to see ex-COVID, the immune response begin to pick up, very quickly. And then in the naive, you can actually again see the day 7, you can see that peaking. And then you can actually see in the ex-COVID and naive day 7, you can actually begin to see that it's now the immune response is quite, significant. And in naive, it's actually peaking. And it's interesting to note that there are individuals who don't seem to have proper immune response to this vaccine at all. And for us, really, this is an issue that we want to be able to understand what is going on in these particular individuals. But it is interesting enough that on day 28, everybody's way up. So there's a dynamics to it that there are individuals where the response is actually very slow, very slow. And then beyond day 28, 29, it picks up actually very, very well. And hence the reason why we are actually beginning to say that maybe don't boost at day 28, boost much, much later. Because this the dynamics here is that the vaccine is actually still in the trajectory state. And you actually want to boost when you have actually reached the peak. Next slide. And we are also just interested in looking at issues of vaccines and different age groups. There's a lot of discussion, about, parasitic infections in our environment, about exposure to various antigens. And we are also being told that immunosenescence in the African setting begins much, much earlier, so that with age, we may not be able to actually respond very well to various immunogens. So we were looking at first dose for those who are below 40, and then those who are above 40. So if you look at first dose here, you can actually begin to see that as time goes on, below 40 first dose, and then you look at 7 days, and then you look at 14 days, and then you look at 20 days, you can actually see that age doesn't seem to really matter much. Again, if you look at those individuals who are naive, again, you'll actually see that there's really no difference between those who are below 40 and actually those who are above 40. But the numbers are not enough. We actually need additional individuals to volunteer so that we can actually segregate this even further to look at further age groups, because saying above 40 is not enough. We want to be able to segregate into even different age groups, maybe 20 to 40, 40 to 50, and then 60 to 70 and the like. But that will only happen if we actually have enough numbers. So this is just a peak, and this is actually only looking at neutralization antibodies in terms of titration. But what we are going to do further with this data is then to follow it up with neutralization. So even if somebody has high levels of antibodies, we want to find out what is the quality of these antibodies in terms of the ability to neutralize the Wuhan strain, as well as to neutralize the various variants that are actually emerging. Then we are also going to go further and also look at the T cell arm, to look at T cells in the same way, to look at naive individuals and also look at COVID individuals in terms of understanding the nature of the T cells and how able are they in terms of cytotoxic ability to actually attack and actually be able to lyse virally infected cells. So this will be only the 2 slides I'll show for now, but as we go on, as we analyze our data and more information comes in, we shall keep adding on to this. So I will say thank you, and then we can go to question and answer.
Thank you very much, Professor Nzala and your team for the good work you're doing. Before I hand over to Robert to coordinate the Q&A section, I have several questions. The first question is to Dr. Marianne. What was the basis of the WHO statement questioning against mixing of viruses? Then to Prof, just ask all of them so that they are responded to together because we have limited time. Is Prof, what is your suggested, because of following the work that you are doing, and I know probably it's preliminary, what's your suggested dosing regimen for Kenya with the current work that you are doing, and what's your opinion about mixing currently? Because I know that's a question in everyone's mind. Thank you.
Thank you, Dr. Thuranira. So again, why WHO asked that question or gave that cautionary precaution about mixing and matching is because we do not have enough data In fact, as I mentioned, the presentation, we only have 3 or 4 groups in Europe that have actually started looking at mixing and matching. So again, I believe the more we get data, the more we see that it's safe and immunogenic enough to mix and match different models, then eventually I believe WHO will then, using data, using science, give that go ahead that indeed we can mix and match. I believe that was just a cautionary, a caution from them. Thank you. Over to Prof.
Yes, in terms of dosing, actually the dosage that has been suggested that we are using for AstraZeneca should remain. Really, there's no problem with that. The issue is schedule. The schedule, as suggested, day 56 to us would actually be totally ideal, because part of it is that you also don't want to delay fully vaccinated for a very long time. And the mix and match where we have received AstraZeneca, if we have Moderna or if we have Pfizer, that would actually be a recommendation. Any of them can actually do. The reason why you see maybe AstraZeneca followed by AstraZeneca being muted as compared to AstraZeneca to Moderna or AstraZeneca to Pfizer is because when you are giving a viral vectored vaccine, there is also immune response to the vector itself. All right. For example, for the AstraZeneca, there's actually immune response to the ChAdOx, which is actually the vehicle. So when you come in with a booster using the same ChAdOx again, the immune vaccine that has been generated towards ChAdOx can easily mute your boost. And that's why you saw the study we did with Jensen and Jensen, we were using AD26 as a prime and MVA as a boost. 2 different vehicles. One was an adenovirus and the other one was modified vaccinia ankara. So I'm actually not surprised that ChAdOx compared to ChAdOx Moderna or ChAdOx Pfizer, the mix and match are actually much better. And that is what informed us, for example, during the Ebola vaccine we were doing, we never used the same recombinant twice. We actually just used adeno26 and MVA as a boost. Over to you.
Thank you. Thank you so much. Robert, kindly introduce yourself. There are so many questions in the Q&A. We have at least 10 minutes to wind up.
Okay, thanks, Doc. This is Dr. Robert Langat from CAVI. I'm a scientist in CAVI together with the panelists today. Thanks for the nice presentation to all the panelists. There are quite a lot of questions. I've tried to answer them live as the presentation was ongoing. So there are some questions that are more clinical, and I would like Professor Nzala to tackle some of them. One from Dr. Peter Ongwae is asking about AstraZeneca and JJ COVID vaccine have shown to cause rare blood clots. The mode of action of these adverse effects has been identified. So he's asking, with this knowledge, how much is being done to develop safer versions of these vaccines to assist address these vaccines' efficacy and reduce mortality?
The whole issue here is that this AstraZeneca that we have used was the first generation. And always vaccines go through the first generation and the second generation. What I know is that the colleagues who actually developed the ChAdOx vaccine, the ChAdOx vaccine, are actually our colleagues in Oxford. We have worked with them and we continue to work with them. They have actually embarked on the second generation vaccine. What is happening now, the immunogen has actually been developed, but now it's still in mice studies. So definitely with that mechanism being known, it is actually clear that a new generation of AstraZeneca will actually be coming through very, very quickly. But also refer to the slide we showed where there are so many vaccines undergoing phase 1, phase 2, and some are actually still experimental. Some of this information is actually being fed back to ensure that better and newer and more safer and more immunogenic products are actually brought forth. Over to you, Langat.
Yeah, thanks, Prof. Yeah, the questions are coming thick and fast. There's Geoffrey Kasambelu who is asking, between mix and match and same vaccine, which one would you advise when it comes to immunogenicity and immunity from these preliminary studies?
Okay, maybe Daniel can answer that. Daniel?
Yes. Sorry, Robert.
Yeah, so he's asking about the mix and match and same vaccine. Which one would you advise when it comes to immunogenicity and immunity from these preliminary studies?
I think all combinations of mixing and matching, whether you start with the AstraZeneca or the vectored vaccines and boost with the mRNA vaccines, or whether you do it the other way around, it looks okay. And that is mainly because of what Prof has described about the anti-vector immunity. So I think the data is very limited, and there's very little we can actually say. We can only speculate that any mixing and matching, as long as it's across different platforms, as long as it's not the same type of vaccine, will be beneficial.
Yeah, also just to note that the Johnson & Johnson, the AD26 is a single dose, but chances are as we go ahead, we might actually find that we might actually boost with an mRNA, or we might actually boost with ChAdOx, because the AD26 in J&J is actually human, while the ChAdOx is actually a chimpanzee. So they are different vectors. But really, this is only going to be informed by science. Over.
Yeah, thanks, Prof. and Dan. Follow-up to that is, Philista is asking, will the mix and match lead to resistance and mutation? Dan?
Actually, the mixing and matching is what will prevent resistance and mutations. Because as Mariana has shown, the mixing and matching is giving better immunity across variants. And therefore, chances of people who have been immunized getting infected by new variants and allowing those variants to evolve further will be minimized. So it is actually, yeah, as shown here. So if you look at all those, Marianne, if you allow, those are different variants as labeled at the top there, B1, 7B, 1351, and P1. And you can see that the mixing and matching is improving responses to the variants. So it will be beneficial.
Yeah, follow up on that slide there, which is on the screen. Martin Sirengo is asking, I noticed the mixing of vaccines data share all begin with AstraZeneca vaccines, then the mRNA. Do we have data or studies that begin with mRNA, then AstraZeneca? Those are like the reverse mixing.
You see that the issue of AstraZeneca, this is historical. It was not actually planned. And a lot of Europeans had already received AstraZeneca when those thrombotic events occurred. And also when they stopped, then the whole issue was that, what do we boost with? And hence the reason why you find that in majority of those countries, it is AstraZeneca first, followed either by Moderna or Pfizer. But I know they are now going to to purposely look at the reverse, because those will have to then be planned. They will have to be go through ethical approval, and those studies can actually be done. But what you are seeing is actually what I would say real world events that actually informed boosting without actually going through. But it was actually based on some of the work we have done before with HIV as well as with Ebola. Over.
Thanks. Someone is asking Donald Sokoti, do we have a special vaccine for people on drugs such as sirolimus to prevent organ transplant rejection? Prof?
Special what?
Special vaccine for people who are on drugs, such as sirolimus, to prevent organ transplant rejection.
No. I think studies are being done because this is a whole issue of comorbidities. People with cancer, people with immunosuppression. We have even data is now beginning to emerge about vaccines in people who are HIV infected. So really there is no one special vaccine. What will happen is that these vaccines that are currently available will actually be tested for safety and immune response in these particular individuals. So for now, I cannot tell you that there's one particular vaccine that is earmarked for those kind of individuals. Over.
To add in, Prof. and Robert, there's a paper that has just come out actually an hour ago, Nature Medicine, where they were doing a mixing of the Chadox, the AstraZeneca, followed by a boost of the mRNA, and they looked at a cohort of people who are immunocompromised, who are also on getting transplants and had malignancies. And they showed that the people who had the mixed approach had a much stronger immune response compared to the ones who got a matched in this cohort of people who are immunocompromised. So in fact, this paper was advocating if immunocompromised or undergoing transplantation, maybe a mixed approach because it stimulates different parts of the immune system and eliciting. So again, it's really hot from the press. Thank you.
Yeah, thanks, Marianne. We have like 1 minute or so to go. There is a comment from Dr. Kidanga. It is known that different populations have different gut microbiota that may possibly influence antibody response post-vaccines. Other immune modulating toxins like aflatoxins have been suspected to influence antibody response. So he's asking, what is your comment in terms of these aflatoxins, hepatitis B that may influence the antibody response after vaccination?
Actually, just go to the last slide. David, the reason why we are conducting our own this aspect in our own population, just the previous slide, is really to try and understand our own immune responses in our own setting. Because as I told you, we are actually plagued with parasitic infections, most of them that impair the gut, and where that's a microbiome. And the microbiome has a big influence on immune response. The food we eat is loaded with aflatoxin. And you recall the work we did is that aflatoxin has some sense of muting the immune system, the whole issue I talked about, immunosenescence. So we will only understand this if we do these kind of studies in our own. So we are actually just piggy bounding on what the country is doing and enrolling individuals. So what I will say is that we are only going to get there if a number of individuals volunteer to come to CAVI and be part of this. As we begin to look at children, we shall also bring in children and maybe collect more information who has additional comorbidities and things of that nature. So David, you are right. We must understand our own environment in order then to inform us what will be the best type of vaccine, the best dosage, and even more so, the best schedule. Over.
Yeah, thanks, Prof. We have been added 4 more minutes. So Keva Bosire is asking, are there any studies that give a suggestion to the period that one gets immunity after complete immunization? And are there individuals that have shown failure to elicit immune response after vaccination?
Now, Daniel, you want to give it a try? Then I come in.
Yeah. So there are some follow-up studies that have happened mainly in the West, looking at the longevity of immune responses after these vaccinations. And actually all the studies that I've seen reporting that, they have shown that immunity is sustained These studies are studies that have taken around at most 8 months. The majority is around 6 months. So it will be interesting to see whether after follow-up of more than a year, whether the immunity will wane. But again, that is complicated by the emergence of new variants. As you have seen in some of the slides, that when someone elicits, when a vaccine elicits an immune response, that response might be very good for the initial variants, but then the neutralizing ability of that person's serum is much lower for newer variants. So that interaction has not yet been evaluated, but generally the longevity of the antibody responses has been shown to be quite impressive, and it will be interesting to see how it goes.
Yeah, and then just to add that, to add to what Daniel is saying, really for us, the dream vaccine should not only concentrate on the crown jewel, which is the spike protein. The vaccine should also look into other SARS CoV-2 proteins, because when you look at T cells, the kind of epitopes that are expressed on infected cells that are actually seen by T cells, they come from a variety of jewel. And hence, if we really want long vaccines, will actually be a combination like what we are talking about here, the whole virus, where you actually bring in additional epitopes in addition to epitopes to the crown jewel, and that is the spike protein. So that is actually the aspect that is going to emerge in the next generation of vaccines. Over.
Yeah, thanks, Prof. And Dan, the last, we have 2 minutes or so. Okwido is asking, will a third dose be needed, or might vaccination be an annual exercise like the flu vaccine?
Marianne, what do you think? Daniel, what do you think? Before
well, this is
what are your thoughts?
It's an ongoing conversation because in the UK, the over 65s are actually being advised to go for a booster. That's a third dose when they go for their flu vaccine in preparation for the winter. Again, we don't have enough data to show the benefits of this third dose. So maybe as the exercise continues, we'll be able to see that. There's an ongoing study, the COMCOV, that's been done by the University of Oxford, where they're studying the effects of giving a third dose. We even have colleagues in the Imperial College who we partner with who are participating in this study. So we'll be able to get this information firsthand and be able to advise if a third dose indeed would be necessary. Over to you.
Anything, Daniel?
The only thing I can add, thanks, Marianne, is that that is very much dependent on the decay of the immune system. It's an interaction between the decay of the immune system and the emergence of new variants. So it's quite complex and it's difficult to predict as of now.
Okay, and then for me, the conclusion is that we are going to come back to this webinar to discuss how is this pandemic going to end? How is this pandemic going to end? And how shall we recognize the end? And how will the end actually look like? So we are going back here. I didn't ask that because the question is that, is SARS CoV-2 going to be endemic? And if it is going to be endemic, in what form is it going to be? And will it require, the way you are asking, regular boosters every so often? And if it is, what boost are we going to give? So just for this space, we are going to come back again as a group and then discuss how will the end of this pandemic look like. Over to you, Langat.
Okay, thanks, Rob. I think that's a good ending. We have run out of time, so I would like to thank all the panelists today and the participants. There have been a lot of queries on the chat box, so we'll try to answer them and share with Dr. Turanira for him to share in the website. So I want to hand it back over to Doc to talk for us.
Thank you so much, Dr. Langat. And before I give everyone a chance to give a closing remark, I have two pressing questions. There's one who has asked, several people have asked, how can they be part of this cohort study being done by KAVI? Maybe you give us the details of how, and then, yes, that one.
Langat?
Yes.
Actually, what you do simply, simply walk to KAVI and then for Dr. Langat. Dr. Langat is the one coordinating, so just walk to KAVI, look for Dr. Langat. We shall go through a process, explain to you. And Dr. Langat, you are there, you want to add something?
Yeah, I just wanted to say, so the cohort or the study that we are studying at the moment are individuals who have been given the vaccine. So if you have been given the first jab, you are welcome to join. Also, if you have got the second jab, you are welcome to join. Also, if you are going for that, for jab, whichever time for the first dose, you can always come and we do the baseline before you get the jab. And then we'll advise you more on the logistics as you go along. So everyone is welcome so long as you are being given the jab.
Thank you very much. Daniel, closing remarks?
Dan, it seems to be muted.
Oh, okay. Thank you so much for giving us this opportunity to present this data, because there's a lot of data a lot of studies are coming out, and it can be a bit confusing if these data are not synthesized. And that's what we are trying to do, to just synthesize the information as it comes out, to make it as simple as possible so that it is understandable. And it will be very interesting to see the kind of information that will come out in the next couple of months. So I really look forward to our future discussions on this topic. Thank you.
Thank you very much, Dr. Marianne.
Very much, Dr. Doranera, and the panelists, and as well as our audience. This has been very informative discussion. And again, we'll keep looking at the science, the data that's coming, and also analyzing our work at KAVI real time. And I'm hoping that we can have many of these forums where we can share this information. Also at KAVI, we have a bi-weekly journal club, again, where we go through our work, our information, and probably Dr. Doranera, I'll give you the link so that you can share with the audience so that they can join us in terms of discussing science, not just COVID science, but other infectious and non-communicable diseases. So thank you very much, and back to you.
Thank you so much, Professor.
Yeah, Doranera, just to say thank you to KNH for giving us this opportunity to discuss with colleagues the status of science, where we are with various things. So let's keep at it, let's be informed by science, but at the same time, we also want to build our own data. So I'll encourage as many as who can to actually walk over to KAVI and participate in this longitudinal study, in where we're actually trying to understand the immune responses. Even when we begin to mix and match in our own country, we shall also try to also ensure that we gather enough data. So with that, I say thank you, KNH. Thank you, the participants, for staying with us this late. It's almost 9. Back to you, Doc.
Thank you. So on behalf of Kenyatta National Hospital, University of Nairobi webinar team, I want to thank the 875 colleagues who logged in. It was nearly maximum. And thank you to the panelists. At this point, I want to wish you a good night and enjoy your dinner. Thank you so much. Good night. Goodbye.
Thanks, everyone. Good night. Goodbye.